So today we will discuss about the topic structure of skeletal muscles. Okay. As we know that the bone is attached with the muscle through a connective tissue known as tendon. That is, tendon is the connective tissue which connects the muscle with the bone. This is the muscle. As we consider the whole muscle and dissect it in a horizontal plane, then we see there is a connective tissue covering known as fascia. That is, fascia is the connective tissue covering which covers the whole muscles from outside. And the outermost fascial or the outermost connective tissue layer is known as epimysium. That is, epimysium is the outermost connective tissue which covers the whole muscle, and it unbundles, it engasps the number of muscle fibers known as muscle bundles. ठीक है. Muscle bundles are also known as fascicles. There are about 10 to 100 muscle fibers or muscle bundles, sorry. There are about 10 to 100 muscle bundles which are separated by a inner connective tissue layer known as perimysium. Muscle bundles contain a number of myofibrils or muscle cells, and each muscle cell is covered by a layer, a third connective layer known as endomysium. When we magnify one myofibril or one muscle cell, we'll look inside it and we found that there are number of nucleus. That is, it is a multinucleate condition. We simply call it as syncytium. Syncytium is that type of multinucleate condition which is formed when karyokinesis is not followed by the cytokinesis. A muscle fiber contains a number of mitochondria, sarcoplasmic reticulum especially. Sarcoplasmic reticulum is the storehouse of calcium here, and it plays an important role in the muscle contraction also. Then if we look into the cell or one muscle fiber, it shows striated bands, alternated striated bands of dark and light. Then one light band is followed by dark band, and then dark band is followed by light band. Then this condition in a progressive way. Light band is formed by actin filaments, and dark band is formed by myosin filaments. That is, if we look into the proteins of muscle fiber, or we can categorize the muscle fiber proteins into mainly into two categories, that is actin and myosin. First we'll discuss the I band, that is light band. I band is that band which is formed by the actin filament which are connected through a line known as Z line. Z line is nothing, it is also a filament, it is also a disc of protein which is formed by the actin itself, and it enlarges in the center, or it gets attached with the actin filament through a protein known as cap Z. We simply say that the actin filament from both the sides, from right or left, is joined with a Z disc through a cap Z protein. Actin filament forms the light band, which we also call as I band, and in the center there is a band which we also call as dark band. Dark band contains the centrally located myosin filaments and its overlapped region with the actin, and also the non-overlapped region. The non-overlapped region of the myosin filament is known as, or it forms the H zone, and the center of the non-overlapped region is connected by a protein which is known as M line. M, as it lies in the middle of the sarcomere. Then the part between the two consecutive Z lines is known as sarcomere, and it is the functional, sarcomere is the functional and structural unit of a muscle. Then we discuss about the actin and myosin, structure of actin and myosin. If we look deeply into the structure of actin filament, these are made up of three structural proteins. First is globular actin, nodules having a site in the center for the binding of myosin. First is actin, then the second is array of tropomyosin filament, and third one is troponin. All these globular actin, troponin and tropomyosin form the filament of actin in such a way that it looks like a spiral. Actin had the site for myosin attachment, as we draw here, and this site in a resting state is covered by the troponin, and the position of troponin is stabilized by the tropomyosin filament. That is, in resting state, the troponin and the tropomyosin covers the myosin binding site on actin. That is, I repeat it again, that is, in actin, the myosin binding site is covered by tropomyosin, and the position of tropomyosin is stabilized by the troponin only in resting state. Troponin has a site for calcium binding. We'll discuss in our next lecture, that is troponin having a calcium binding site. Now if we discuss about the myosin is polymerized. It is a structure formed by the polymerization of small proteins known as myromyosin. We can say that myosin is formed by the polymerization of small polymers or small proteins known as myromyosin. And if we look into the structure of myromyosin, it has a globular head and a long tail. The globular head has the two sides. One is for actin binding and one is for ATPase activity. The globular part of myromyosin, that is this one, the globular part of myromyosin is known as head, and the thin elongated part is known as tail. So head having the activity of ATPase, that is, it hydrolyzes the ATP and convert it into ADP and inorganic phosphate, or it attaches the ADP with the inorganic phosphate and convert it into the ATP. Light band is formed by the actin filament and is also known as I band, and actin are thinner filaments, and dark band is formed by the myosin filament and is also myosin is also known as thicker filament. We'll repeat it again. The different zones of the sarcomere. Sarcomere is the function unit of muscle fiber containing different zones, that is I band, A band, and then alternative I band. The alternative I band gives the muscle fiber its characteristic appearance, that is striated appearance. First light, then dark, then light, then dark, then light. And the region between the two consecutive Z line is known as sarcomere. Z line connects with the actin filament and it also connects with a protein known as titin. Titin is the longest protein of human body. Titin is the longest protein of human body. It actually connects with the M line in the center and the Z disc in the periphery. So during muscle contraction, it shows a characteristic function or stabilizes the M line in the center. We'll discuss it in our next lecture. This is all about this overview of structure of skeletal muscle. Thank you very much.